Hello, dear student, class 11. I am going to start the topic glycolysis. What is glycolysis? Glycolysis is a part of respiration. It was discovered by three scientists named Emden, Meyerhoff, and and Parnas. That is why it is called EMP pathway. EMP pathway. It takes place in cytoplasm, so that is why it is also called cytoplasmic respiration. It is called cytoplasmic respiration. It is common in both anaerobic or aerobic respiration. So that is why it is called common respiratory pathway. So it is also called EMP pathway. It is also called cytoplasmic respiration or common respiratory pathway. What is the site of the glycolysis? It takes place in cytoplasm of the cell. What is the definition of glycolysis? It, it is a multi-step process. It is a multi-step process takes place inside the cytoplasm in presence of enzymes. Multi-step process. Each step is controlled by enzyme. Important flow chart of the glycolysis represented as below. Glucose and glucose combined with ATP to form glucose 6-phosphate and ADP, and it is mediated by an enzyme called hexokinase. Glucose 6-phosphate later on converted into fructose 6-phosphate. Fructose 6-phosphate further combined with ATP to form fructose 1,6 fructose 1,6 diphosphate. In presence of enzyme phosphofructokinase, fructose 6-phosphate in presence of aldolase enzyme form two triose unit called GAP and di-HAP. They are isomer. Glucose is converted into glucose 6-phosphate in presence of hexokinase enzyme. Glucose 6-phosphate is further converted into fructose 6-phosphate called phosphofructoisomerase. Phosphofructoisomerase. So isomerization process takes place. So name of enzyme is called isomerase. Fructose 6 phosphate is further converted into fructose 1,6 diphosphate in presence of enzyme phosphofructokinase. Glucose is a 6 carbon compound which is converted into 2 triose unit. They are called 3 carbon compound, 3 glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. Both are isomer. Glyceraldehyde 3 phosphate contains aldehyde group while dihydroxyacetone phosphate contains ketonic group. Glyceraldehyde 3 phosphate further forms 1,3 diglyceric acid or bis-phosphoglyceric acid. Phosphoglyceric acid. In presence of NAD plus NADH2 or H plus H plus. Glyceraldehyde 3 phosphate is converted into 1,3 bis-phosphoglyceric acid in presence of an enzyme called dehydrogenase. Dehydrogenase enzyme is responsible for the conversion of phosphoglyceraldehyde into 1,3 bis-phosphoglyceric acid, then converted into 3 phosphoglyceric acid. Three phosphoglyceric acid to form ATP. Here ATP combined with 1 phosphate to form ATP. Further, 3 phosphoglyceric acid is converted into 2 phosphoglyceric acid, 2 phosphoglyceric acid in presence of enzyme mutase. In presence of enzyme triose or PGA kinase. It is a multi-step process, number 1, number 2, number 3, number 4, number 5, number 6, 7, 8, 9. Further converted into PEP. PP is also called phosphoenol pyruvate. And last product is called pyruvic acid. Pyruvic acid. This is a 3 carbon compound. Pyruvic acid is a 3 carbon compound. And here 1 ATP is produced. ADP combined with IP to form ATP. So it is called PP kinase or pyruvate kinase. Enzyme is required for this conversion. Pyruvic acid, the formula of pyruvic acid is CS3, CO, COH. And the molecular formula of glucose is C6H12O6, which is a 2-carbon compound. So net reaction of glycolysis is that glucose is converted into 2 molecules of pyruvic acid, 2 molecules of pyruvic acid with the release of 4 atoms of hydrogen, and energy is also released. The molecular formula of pyruvic acid you know very well, CH3COCOH. So this is a 3-carbon compound, while glucose is a 6-carbon compound. The molecular formula of glucose is C6H12O6, and each step of glycolysis is controlled by enzyme or enzyme mediated step. Glycerol 3 phosphate is converted into dihydroxyacetone phosphate. Here isomerization process takes place, so it is mediated by isomerase enzyme. Next step, dehydrogenase, PGA kinase, mutase, enolase, and P kinase, pyruvate kinase. So ultimately pyruvic acid is produced. How many energies are? What is the energy output of glycolysis? So energy output, there are net gain of 2 ATPs. ATP consumption site, ATP consumption site. At 2 site, that is step number 1 and step 3. 2 ATPs are consumed. 2 ATPs are consumed, while 2 ATPs are produced by GAP and ultimately by pyruvic acid. Dihydroxyacetone phosphate also responsible to form pyruvic acid. So that is why there is net production of 4 ATP. So while net gain of ATP is equal to 2 ATP. They are called substrate level phosphorylation because this ATP is produced directly from the substrate. Besides the production of 2 ATP, 2 NADH2 are also produced. NADH plus H plus or NADH2. But NADH2 is not available during the anaerobic process. So they are available in the process of aerobic respiration. So NADH2 are unable to produce ATP during the anaerobic respiration. As already told, glycolysis is a common respiratory pathway. So that is why it is common in both anaerobic as well as aerobic. In anaerobic glycolysis, only 2 ATPs are produced. But in aerobic respiration, 2 NADH2 also undergoes electron transport system, and as a result, total ATP is produced in aerobic respiration in part of glycolysis is 6. So that is why aerobic respiration is better than that of anaerobic respiration. Thank you.